Welcome back to the micro-module series, Understanding Bias in Epidemiological Studies. In this module, we will be focusing on two methods for controlling confounding, which are restriction and matching. The objectives of this module are to explain how restriction can help control for confounding, explain the limitation of restriction, explain how matching can help control for confounding, and explain the limitation of matching. Let's talk about restriction first. Restriction means to restrict the study or analysis to one category of the potential confounder while excluding others. In other words, only the individuals with certain characteristics are included in the study or analysis. Again, let's look at the previous example of smoking and lung cancer, where the potential confounder, age, has two categories, young and old. To prevent the confounding caused by age, we can restrict the study to young people only and exclude old people. As a result, in both the exposed and non-exposed group, 100% are young people. There is no longer imbalance in age between the two groups. Thus, age will not cause confounding. The major limitation of restriction is that if there are more than a few restricting factors, and each of them represents a fairly large proportion of the population, this approach will exclude many people, making it difficult to achieve the desired sample size for the study. Also, the generalizability of study results will be undermined, as the study subjects do not represent the general population in terms of the restricting factors. For example, consider a study that is restricted to young, non-smoking female participants. As old people, smokers, and male are excluded in the first place, and each of them represents a large number. Only a small proportion of the population is left for recruitment, and many of them may not want to join the study. This will make it more difficult to recruit a sufficient sample size than in the case without any restrictions. Furthermore, the study results will be applicable to young, non-smoking females only because they are derived from a population with these characteristics. The second method for controlling confounding is called matching. In cohort studies, matching refers to the selection of unexposed subjects that in certain important characteristics are identical, or nearly so, to the exposed ones. In case control studies, it refers to the selection of controls who are matched with the cases in some characteristics. For example, in a cohort study examining the association between smoking and lung cancer, we might include 15 smokers and 15 non-smokers. For each smoker, we would find a non-smoker of the same sex to serve as a match control. By matching smokers with non-smokers based on sex, we can ensure that the sex distribution is the same between the two groups. As there is no imbalance in sex, this factor will not cause confounding in this study. The major limitation of matching is that if the factors to be matched are more than a few, the feasibility of study would be severely undermined. For example, if you want to match each smoker with a non-smoker on 3 factors, namely sex, age, and educational level, it would be very difficult to find a sufficient number of non-smokers that fulfill all these criteria and are willing to participate in the study. Okay, in this module, we discussed how restriction and matching can help control for confounding and the limitations of these two methods. In the next module, we will talk about the use of standardization for controlling confounding. Thank you for listening.